Good morning, everyone. Okay, so today we're going to be starting our last section. And this section is going to consist of kind of one big subject, which is control of gene expression, right? So we've talked a lot about gene expression as a process, right? But really we want to now talk about, okay, well, how is that controlled? And then towards the end of that section, we've got a few other kind of fun topics to talk about, like biotechnology and transgenic organisms and things like that, right? So we'll kind of wrap up by tying all of these things that we've learned together in some kind of practical applications, like how is this stuff used and things like that. And so really the big deal about the control of gene expression is that it's how we are who we are, right? It's not just how we are as individuals, with our particular predispositions, right? So I don't know if you know people in your family or the like, which are kind of prone to getting addicted to stuff, right? Not necessarily talking about like hard drugs and stuff like that, but I don't know, collecting picture cards or collecting seashells. Like one of my daughters is nuts about collecting seashells. Just collects bags of them and never does anything with them, right? So kind of those particular types of behavior, you can see them go through families, right? So there's clearly a genetic component to that, right? And um, how can I put it? Obviously, there's a large part of environmental uh, part of that too, right? So it's really hard to kind of disentangle stuff like that in human genetics. But the reason why we have all these complex behaviors is this very, very complex interplay between what our genes encode, right, which is the actual sequence of those genes, right? So, and that changes between individuals based on what alleles you inherit, and also how those genes are expressed. And that is actually uh, one of the clearest paths by which the environment can affect phenotype, right? So if you're in a very hot environment, you're likely to start turning on genes involved in protecting against heat damage, called the heat shock response. If you're, in a very dry environment, you're likely to start increasing the expression of proteins called aquaporins in your kidneys, which are then able to reclaim more of the water uh, from, pardon me, your urine, essentially, to the point where actually, I don't know if any of you have experienced this, but I have on kind of long bike rides in hot and dry places, you don't actually pee, right? Which is kind of crazy. You don't produce any urine at all, even though you can be drinking, gallons of water. So anyway, so that's the reason why we talk a bit about, mutations and stuff like that, but really at the end of the day, it's how those genes are controlled, which makes us who we are. And it's also what makes us different to closely related species, right? We are very similar in a lot of ways to chimpanzees, but obviously we're different as well. But we have almost exactly the same DNA as chimpanzees, kind of like 99.9% identical, right? I think, or 99.1%, something like that, right? There's not a lot of difference in terms of DNA between us and chimpanzees, and yet we are clearly very different in many ways. Right? Also, one of the reasons why talking about control of gene expression is so interesting is it's not just about kind of the here and now, like why are we the way we are now, is also why are we the way we are in an evolutionary sense. And so part of our evolution has been the accumulation of changes in genes or even duplication of genes, right? There's a whole gene family called Hox genes, which control kind of essentially patterning along your body, right? cervical vertebrae are not thoracic ones because of Hox gene expression, right? You can imagine it'd be pretty terrible if you had ribs coming out of your neck. So anyway, that gene has gone through, oh, I don't actually know how many duplications. There's 7 different clusters, and there are up to 15 different genes in each cluster, just to give you a rough idea, right? So obviously that's part of our evolution, but also the gain of the ability to control a particular gene in a particular way or in response to a particular signal is also a big part of how we've evolved, right? And so a large part of the complexity of us as humans, as we have evolved over the millions of years, is not down to the differences in our protein coding regions, right? Again, those make up a very small part of our genome. We have approximately the same number of genes as chimpanzees and fruit flies. Actually, I think the fruit flies have a little fewer. Soil nematodes, and so on, right? But it's how we control those genes that matter. And that's why this particular section, I think, is so interesting, because it impacts everything that we do on a daily basis. You know, how much sugar did you have in your coffee this morning, right? That's going to change the expression of insulin, right? Which in turn changes the expression of other things, right? Which deal with that extra sugar going into your blood, right? For example, right? Even on a daily basis. But also it's kind of like big picture stuff as well, right? So essentially what we mean by the control of gene expression, I'm just going to, we've got this in the PowerPoint too, but I like to draw this out because it's makes things a little clearer. And so, man, I really write terribly. There you go. And this is, I mean, there are some very major differences between prokaryotes and eukaryotes in control of gene expression, which is why we've separated them into kind of 2 distinct subjects and 2 different PowerPoints. But the overall aim is that we need to go from DNA, which is our info, to functional protein. So far down there, you can't even see it. Let's stretch this back a little bit. Right. And so we've got to go from this to this, and we need to control how much of this we have and where it is expressed and what is it expressed in response to, right? So there's no point making a shit ton of insulin if your blood sugar is stable and you don't go on crazy sugar binges, right? It's just a waste. It's actually a really bad thing to do as well because it stresses out the cells that make it, right? That's one of the reasons between why type 2 diabetes happens is that essentially your pancreatic beta cells just get worn out from producing so much insulin, right? So we have to control this very, very, very carefully, right? We also don't want this being produced when it shouldn't, right? So if we have a pathway which is involved in controlling development, you don't want that signal to come on when you don't want that particular developmental program to occur, right? That's essentially, in many ways, a bit of a simplification, but that's in many ways the start of cancer, right? Is this production of a signal telling the cells to grow when they shouldn't. Okay, so essentially we've got this big gap between the two. We've got our information up here, and we've got our functional protein, which essentially does stuff. Push that cushion out of the way a little bit more. And so we need to get this into not turn this into this, but take the information from that to create this. And so in many ways, right, it seems simple, right? Our steps between DNA and protein, actually, let's do one long line. We'll change that in a little bit. Is this, right? You know, the sequence hypothesis. DNA gets transcribed into mRNA, which gets translated into protein. All well and simple. Well, when do we produce this, right? So we have control of transcription. Here is very, very important. You also know that we have a whole bunch of other stuff going on before we get our mature mRNA. We're not talking about our pre-mRNA here. We're talking about mature mRNA. So we also have control over mRNA processing, particularly splicing. Man, what is going on with my writing today? Just does not seem to work. Trouble taking a weekend off. For example, splicing, right? So how you splice that mRNA has a big role in what mRNA you get out at the other end. Okay, so obviously that's a eukaryotic deal, right? Because prokaryotes don't process their mRNA. Then we have this stuff, right? So this has to get exported out of the nucleus into the cytoplasm, and then it has to be found and translated, right? So if we get rid of that mRNA, we don't get any translation, we don't get any functional protein, right? So we have mRNA stability, which is a huge part of control of gene expression. Because if you don't have any mRNA, you don't have any protein produced from it, right? And stop me at any point if any of you have any questions as well. So actually what we have in here is protein. No, I haven't put functional protein in the front of that. That's for a good reason. Right, before we even get to that though, and again, this just shows you how astonishingly complex this whole process is. We also have control of translation. Right, we talked about that some in terms of particularly eukaryotic translation. You know, the role of the 3' UTR and the poly A tail, particularly the poly A tail in translation initiation. Okay, so that's another way in which we can control and manipulate this process. So once we get to having a protein, that's not necessarily a functional protein. And what we mean by a functional protein is the thing that does something, right? It's the one that does stuff. And actually, insulin is a fabulous example because insulin, as it's produced by your... Oh, sorry, I was wondering what was squeaking by the window. As your pancreatic beta cells make insulin, they don't actually make it initially in its active form. It has to go through a whole series of processes before it's functional, right? So we have protein processing is one of those things. We can also control how much protein remains intact. So we can actually tag proteins for destruction, right? If we're done with them, we don't need them anymore, or if they're damaged or whatever, right? And so we also can control protein degradation, right? Which in some ways is analogous to mRNA stability too, right? It's a kind of a negative process, right? Because you're removing something. And then finally, as well, proteins can be modified, right? In actually a surprising number of ways, not just modified so they're degraded, not just modified in terms of cleavage so that they assume an active form, but they can be modified by the addition and removal of things like phosphate groups, right? And so there are many proteins, some of which I've studied in the past, which only work when they've been phosphorylated, right? Transcription factors only enter the nucleus when they've had particular protein residues phosphorylated, right? So there's a final step, which is really protein modification down here. And all of these things can be controlled in one way or another. Typically, when people talk about control of gene expression, this is what they tend to focus on, right? Because that's the obvious one, right? If you don't get that information out of DNA into mRNA, you're not going to get any protein produced anyway, whatever happens to the rest of it. But really, all of these things, essentially, what you have down here is the sum. Like if you think about this as a complex equation of lots of different factors and stuff like that, every single one of these things, obviously depending if you're prokaryotes or eukaryotes, because that doesn't happen in prokaryotes, but every single one of these things adds together, and the sum of that is how much functional proteins you have. Oh, good question, Jelly. No, actually they don't. So typically what happens in, let me take a picture of this. Oh, shoot, where's my camera? Give me a second. Let me get a picture of this first, and then I can clean that off and draw it out, because it's easier to draw stuff quite often than it is to try and describe things with words. So if we're to use insulin as an example, because it's a particularly neat example, essentially you have your insulin gene, then your insulin mRNA, and this gets translated, that's a ribosome. Let's use green. Blue doesn't play very well with my board for some reason. Right, so this is our ribosome in our rough endoplasmic reticulum. Right, and so it's in the rough endoplasmic reticulum because insulin ends up being exported, right? So insulin is something that goes around in your blood. It can't stay in the cytoplasm, so it has to get made by a ribosome in the rough endoplasmic reticulum, right? And so once that happens, you now have, if this is your rough endoplasmic reticulum, right, with your ribosomes all over it. Actually, this had better be going down here, if I'm being correct, if we're in the rough endoplasmic reticulum. So insulin looks something like that, let's say. It doesn't look exactly like that, but that's a pretty decent pass at it. So in this form, this is, I hate working on tiny boards. This is pro insulin. Don't worry too much about that name. But basically what I was telling you is that this is not in its finished form. And then this gets sent to anybody recognize what that organelle is? Stretching back to your Gen Bio 1 days. Yeah, I'm guessing the GA stands for that's it, Golgi apparatus. Exactly. Right. And so as the vesicle containing pre-proinsulin goes through the Golgi apparatus, that's probably better write this up. Essentially, at the other end, you get a vesicle where your insulin looks like this. So it starts like that, then it gets cut, in this progression through the Golgi apparatus until it gets into a vesicle, which is vesicle. And this is your beta cell, essentially. May as well manage to fit this all on one tiny, tiny whiteboard. But still, this isn't functional insulin, right? Because it can't be functional inside the beta cell, because the purpose of insulin is to tell your muscle and liver cells to take up glucose from the bloodstream, right? So really only once it's kind of fused with the membrane and now you have your product outside, ta-da, now you have insulin, functional insulin in the bloodstream. And then that floats around, finds some insulin receptors to bind to on the surface of muscle cells or the like. And then once it is, beta cell. So essentially in the pancreas, you have largely two types of cells. One which is called an alpha cell, which is a, hang on, let me type this down. I'll come back to this in a sec. So alpha cells make glucagon. And that's essentially turning turn glycogen, which is our kind of easy access storage molecule for glucose, right? Can get turned eventually into triglycerides, but they're a lot harder to turn back into glucose or to get back into not glycolysis, but into the Krebs cycle, right? So glycogen is our ready access kind of glucose store when we don't need glucose right away. And beta cells do the opposite. They make insulin, and that tells muscle largely muscle, but also liver as well. So muscle cells don't make glycogen; they use it for generating ATP. But liver cells do turn glucose, generally speaking, into glycogen. Right. So that's what a beta cell is. And so those are the beta cells are the cells that die, usually by apoptosis in type 2 diabetes. So one of the reasons for not the autoimmune type diabetes. Hang on a second, Amber. But certainly the metabolic syndrome type diabetes, where it's from having an elevated blood sugar level for a long period of time, those beta cells die. And that's one reason why you have diabetes, because you're not producing insulin, right? So you're not controlling your blood sugar level. So this is a yes. So this is an example of gene expression, but really particularly an example of the last part of gene expression, which is about taking a protein and turn it into a functional protein, right? And so this is to show that obviously all of this is still important, right? So we have to be able to detect when insulin levels in the beta cell are low so that we can increase the expression of insulin to generate more insulin mRNA to generate more insulin protein, right? So this is still being controlled, right? So this, don't want you to think of all of the control of insulin expression is down to all of this stuff. That wasn't really the point. The point was more in response to Jelany's question about these steps of protein control of protein functionality, essentially, right? And so, kind of getting back to the point, generally, yes, these are typically sequential steps, right? So, and they're actually sequential steps in space as well. So each part of the Golgi apparatus does another modification on the insulin protein, so that we go from this pre-pro to this just flat out insulin. No, exactly. So Amber, this isn't something that happens for all genes, right? This is a specific example of how important protein modifications can be to protein function. So if you took this pre-pro insulin out of a beta cell somehow, and you put that into your bloodstream, it would do nothing. It would be completely non-functional. So it doesn't matter how well you control all of this. If you don't finish the process, you're still not going to get functional protein. And that's why, I wish I could kind of go back to it. You start up here with DNA and you start at the bottom with functional protein, and all of the steps in between added together make up the control of the expression of that gene. So for other genes, say that have proteins that don't need to be processed or don't need to be exported from the cell, none of this applies, right? So it's very much depends on the protein in question or the gene in question, right? What all needs to be done for that protein to end up at the right level, in the right form, and in the right amount, and in the right place, right? So essentially, those are all kind of really important things that have to happen, and they're controlled by multiple different steps, right? And so really, with that previous picture that I drew, not all of that always applies to every gene, right? And it very much depends on what the function of that protein is as to what aspects of its control are used, right? Very much context specific. And that's also one of the reasons why I want to get across how amazingly complex like this whole process is. Because you may have things which are promoting the transcription of a gene, but maybe promoting the degradation of the protein, right? So the overall output is very little protein. Even though you're transcribing the gene up the wazoo, you don't necessarily get a lot of protein out the other side. Right. So yeah, it's very hard to generalize gene expression, Amber, really at all, right? Because it's very much dependent on what the gene is, right? And what the protein is. Do you need that protein in the cell? Do you need it in the cell membrane? Do you need it in the external extracellular space, wherever that might be? How much do you need it? Is it something that you need all the time, right? So, for example, you always need ribosomal protein, right? Because you're always translating proteins, really, unless you're under starvation conditions. And so the genes that encode ribosomal proteins are going to be regulated in a very, very different way to something which is conditional, right? So you need insulin, yeah, more or less all the time, right? But you need to make a ton of it right after you've had a meal because your beta cells have they've kind of like vomited out all of their insulin to help you control your blood sugar homeostasis, right? Or if you are of legal age and so inclined and you have a couple of beers or something like that, then alcohol dehydrogenase will kick in to action and get transcribed and translated and produced in your liver to turn that ethanol into something that isn't toxic, right? But it will never be produced otherwise because if you don't have ethanol in your bloodstream, then like who cares, right? So it very, very much depends on the individual gene, the context of how that protein works, and everything else, right? I mean, it's very much a like hand waving kind of topic because you can't really generalize about any of it, right? It's very much dependent on what gene you're talking about. And that makes it, in a sense, kind of a hard topic to talk about because I can only really give you the kind of general overview and processes, right? I can't really dive into many of the details because we'll just get lost, right? It'll be very much like Alice and diving down a rabbit hole, right? You'll just never come out the other side. So I don't want to obviously scare you all, right? Because that's not the point. I just want to give you like the kind of a look through the window into the complexity of gene expression and gene regulation. And so this is kind of going back to the PowerPoint. And the reason why I like to use that is because it's kind of neat to work through this rather than just having a bunch of stuff up in front of you. But this does a really good job of capturing, right, some of those different questions. Oh, and another great example, right? Insulin is only ever produced in beta cells. It's not even produced in other cells in the pancreas, right? It's not produced in your brain, in your muscle, in your intestine, anywhere but that one specific type of cell. Okay, so every cell in your body has the gene for insulin. But that gene is only turned on in your beta cells, nowhere else. So how does that work, right? How do you get such specific control of gene expression? Another example might be dopamine, right? There are certain neurons in your brain which produce dopamine and respond to dopamine. They're called dopaminergic neurons, right? It would be catastrophic if dopamine was produced elsewhere or detected elsewhere because then you get all kinds of funky signals which you shouldn't get. And so what cells these genes are expressed in is in many ways as important a question as how they're or how much is produced. And so these are just some of the questions, right? They're not all of them, just some of them, right? And it really kind of gets to the point that this process is extraordinarily complex. And so this is essentially capturing some of those different points that I covered in that whiteboard kind of work through. Oh, can I answer them? Whew. Okay, so
Well, let me be more specific. Just for the sake of future exams, you
yeah.
know, we should have a general idea of what the answer to those questions are.
Yes, you should. And ways in
So
which,
what would be like a good example to, what would be a good example of an answer? Because I know it's hard to generalize, but we still need to know it.
right. So this last one down here, how will the gene product be modified and where will it be localized? The example of insulin is a great one for that, right? It's going to be the protein has to be modified and it has to be stored in vesicles to be exported into the bloodstream, for example. If we're talking about dopamine, right? Well, dopamine is actually produced by a series of enzymes, each of which has to be controlled. But dopamine also has to be, pardon me, uh, controlled and released in particular places because it's a neurotransmitter. So you have to turn on those genes that are involved in dopamine conversion only in the neurons that produce dopamine, right? So that kind of answers this particular question. What type of cells would this gene be expressed in? If you think about the genes involved in, let's say, glycolysis. Well, glycolysis occurs in pretty much every cell, I think, maybe red blood cells. No, red blood cells don't because they don't have any DNA, right? So that's kind of like one off to the side. But every single cell in your body needs to metabolize glucose to produce ATP in one form or another, right? So every cell in your body is going to express those genes. But you could answer, I would say you'd probably be fairly confident in saying those genes are going to be more highly expressed in muscle cells than they will be in, let's say, fat storage cells. Does that make sense? Because you have to think about the context of what cells are actively turning glucose into ATP. Well, muscle cells, liver, maybe, neurons, right? So even though every cell in your body expresses the cells and the genes involved in glycolysis, specific cells are going to do that to a greater extent than other cells because of their context, their metabolic needs. Right. This, where will the transcript be localized and when will it be translated? So localized can, particularly in terms of eukaryotes, can talk about is it going to be in the cytoplasm or is it going to be in the rough endoplasmic reticulum? Right, because that determines whether that protein is going to be in the cytoplasm or in the cell membrane or in the extracellular space. And when will it be translated, right? So a lot of organisms actually fill their eggs with mRNAs that are not translated. They're actually inhibited from being translated. So that once that egg is fertilized, instead of having to wait for all those genes to be transcribed, there's a whole bunch of mRNAs just waiting there to produce the proteins that that zygote needs for its early stages of cell division and development and so forth. Right. So these mRNAs aren't necessarily even translated for some time. Right. So those are all the kinds of different questions that you can think about, right? And really, whenever you come across a particular gene, right, you can ask these questions. Well, where is this gene expressed? What is it expressed in response to? When is it expressed in over the course of a day, right? In case of some genes, right, that we have circadian rhythms in terms of gene expression, or over the course of that organism's development. Some genes are turned on very, very early and then switched off forever, or maybe come on later, or whatever, right? There's lots of different ways of thinking about that, right? How much of that product are we going to get? Is it something produced at a constant level, or does it peak and then fall down? What controls that peak and then subsequent, subsidence? Is it control of mRNA transcript stability? Is it control of translation? Is it protein degradation that's getting rid of that protein? What is it, right? So those are all questions that you can ask of a particular gene, especially if you don't know anything about it. Those are the very first things that you would do, right? And so if, for example, project that I worked on as a postdoc, right, was a signaling channel, transient potential receptor vanilloid protein there, TRPV for short. And it was involved in transducing a signal as an ion channel, right? And so the very, very first thing that we did, or I did, was figure out, well, what cells is it expressed in, right? And so I used a GFP reporter, a green fluorescent protein reporter, to see in the worm, oh, it's expressed in those cells and it's expressed in those ones, right? Subsequent work that was done by a grad student figured out that it was a membrane protein, right? Because if you look at the cells really, really close, you can see the cell, and then the protein localizes to the periphery, right? So it's a cell membrane protein, and it's a channel, right? So you would expect it to be in the plasma membrane, and all kinds of other questions like that as part of figuring out, what it does, what's it modified by, and what's the consequences of it working or not working, right? And so that was just an example, right? So I can't give you really specifics, because anytime I give you specifics, I'm going to have to use a specific example of a certain gene, because I can't make those sort of specific statements about other genes because they may not be true. Do you see what I mean? It's very much a context dependent oh, okay. So sorry, I missed my chat thing disappears whenever I click on my PowerPoint. It's not that I'm ignoring you. So essentially, this is a bit of a misnamed PowerPoint, because what we have going on at the beginning up to and including here is very much generic basic stuff, right? So if I get time, I'll go through that today. If not, I'll start that on Wednesday. The rest of this PowerPoint from this point onwards is about prokaryotes. And so, pardon me, prokaryotes do need to answer the same kinds of questions. And in fact, in many cases for prokaryotes, it's even more critical to get this right, because translation, as you know, has a high energetic cost, and transcription too, right? So if a bacterium were to produce something it doesn't need, then it will grow more slowly, it will divide more slowly, and it will basically lose the race against all the other bacteria around it to colonize that particular space, that particular ecological niche, right? So it's very, very important for prokaryotes to control their gene expression tightly. Right? And
okay, for the location question, then just in general terms, you could just like the cytoplasm.
Well, so prokaryotes have a lot of proteins in their cell membranes, and actually they export an awful lot. So, well, also fungi as well. But you heard of a bacterium called Streptomyces? It's kind of one of the common soil-borne bacteria. Not always bad for you, but there's a whole bunch of them. So those that genus is responsible for, I think, 2/3 of the naturally produced antibiotics that we have discovered. I wish I could remember some of them. What is it? Muprosin is one of them. Streptomycin, obviously, and there's a whole bunch of others, right? And so some of those are compounds, some of them are kind of truncated proteins, but that prokaryote has to be able to get that stuff out. So a lot of prokaryotes have proteins in their membrane or in their cell wall. I'm not like super well up on bacterial morphology, right? But they do create, double cell walls. They have like peptidoglycan, which is a kind of a sugar protein kind of mix that helps protect them against osmotic stress, right? So they also have to figure out where in the cell those proteins go, right? And I'm not super familiar with how that's done because in eukaryotes it's really simple. It's right, it's either cytoplasm or rough endoplasmic reticulum. In prokaryotes, I don't actually know. I should look into that or ask my microbiology colleague, right, how that's controlled. But yeah, the same questions still apply for prokaryotes. It's just the context in which they're asked are going to be different, right? It's not going to be what types of cells a gene is expressed in, but what kinds of conditions, right? Under what external conditions does a particular gene need to be expressed in? And if you think about it in a sense, right? So we have, if we think about us as animals, particularly mammals, right? Warm-blooded creatures, we're a lot better at controlling our internal environment in response to our external environment than other organisms are. If you're a bacterium, right, you can't just start shivering or put some clothes on if you get cold. You have to be able to do enact a response that allows you to survive. Same as if they get too hot or too dry or too wet or whatever, right? So if you think about it from the perspective of prokaryote, they actually have to do an awful lot more work in responding to their environment than we do at a cellular level, like on a molecular level, because they can't just go somewhere else or change their biology. They have to actually change what genes are expressed in order to survive and adapt to those changing conditions. So bacteria actually, in many ways, have a harder job of it in terms with respect to the environment than we do. So it's kind of, so again, these questions aren't really eukaryote only. They're very much applied to prokaryotes too. It's just the context in which those questions are asked is obviously going to be different. And there's a whole single cell versus multiple cells kind of deal as well, which kind of ties into that. So at the beginning of next class, we'll start talking about different types of transcriptional regulation. So if you want to get a jump, please do read ahead, particularly about the lac and the trip operon. So these are fairly complex processes. The lac operon is actually much simpler. So this is the lactose metabolism, and down here we're going to have, this is still lac. Trip operon's down here. There you go. And control of tryptophan metabolism is much, much more complex than lac. So don't get the idea that prokaryotes are simple. They're not. They, in terms of gene expression, they are pretty funky, and they've got different ways of controlling it than we do, which is why we're going to go into this in some depth. And I think we've got a couple of days in our schedule. Where's our schedule? Here we go. Yeah, so we've got the rest of this week essentially to go through that. So it's a pretty solid chunk of material to get through, and it can be quite confusing, right? So if you can have a read through of that PowerPoint, check out the textbook if you can, look up stuff in Wikipedia, whatever it takes to give you a better understanding. So when we do cover it, it's going to be a lot easier to kind of get the the more subtle differences rather than just the basic terms. So yeah, Amber, if you got any questions about any of this stuff, please just don't hesitate in asking, because they're all good questions, and they're also really interesting questions too. Right, so I've got to be a little bit careful, A, because I don't want to overgeneralize, and B, because I need to make sure I need to get through stuff. But as best as I can, I'll be very happy to answer questions like that. Anyway, that kind of takes us to the end of the class, and we'll dive into the details on Wednesday. So I'm going to love you and leave you, and I shall see you all on Wednesday. So have a lovely day and see you then.